Okay? So if electrical allows you both ways, you can't really exploit electrical synapse in the situation when you need information being transmitted one way. But chemical synapse allows you to make the signal flow unidirectional. Does that make sense to you? So again, all chemical synapses, all synapses in the nervous system are chemical synapses. Okay, we're good? Is that understood? So let's figure out how the synapse, the chemical synapse. From now on, we're going to talk about chemical synapses. So how it works, what are the features, and then we're probably going to, I'm going to do like a separate, well, maybe we're going to get neurotransmitters in there. So this is going to be, remember on the image of the neuron that I made before, there were like little tentacles at the end, axonal terminals. So this is the axonal terminal of a presynaptic neuron. Now on this side, logically, you're going to have a dendrite of a postsynaptic neuron. We're good so far? Follow? Okay. This get right here, it's called, it's like physically existing, it's called synaptic cleft. So first thing that happens during the synaptic transmission is so-called action potential. Some of you may have heard that term, some of you may have not, some of you may know what it is. We're not going to talk about it right now because it would be just piling too many concepts all together. So just believe me that action potential is the electrical impulse. Okay? And I immediately run into the situation when I need to pause this conversation and talk about something different. Okay? I apologize for that. I thought that I can do it later, but no, I can't. So we're going to chat about it right now. It's going to be very short. You probably know that, so it's going to be just a review. So what I want to review, basically, forget about synapse for now, is transport across the membrane, surprisingly. Without going into too much back and forth, you should remember that the cellular membrane is a lipid bilayer, which does not allow hydrophilic particles, molecules, ions, to go through it directly, like David Copperfield going through the wall, right? They cannot do it, okay? They need, only lipid-soluble molecules can do David Copperfield here. For ions, you need a door, okay? So if they go via diffusion or passive transport, they need a channel, okay? But if you are lipid-soluble molecule like oxygen or carbon dioxide or alcohol, and this is one, like, why people get drunk, because alcohol is very easily crossing all the cellular membranes, can get in the blood very easily, can cross blood-brain barrier because it can go through the membranes directly and eventually get into the brain. Okay? But sometimes, so diffusion, as you may remember, is the process in which particles move along the concentration gradient from the high to low concentration. Does that make sense so far? But sometimes you need to push particles in the opposite direction, against the concentration gradient, sort of uphill. Okay? And this is called active transport. And active transport requires ATP and pumps. This is good so far? It's pretty straightforward. So you can open the nipple on the bike tire and it will deflate by itself. But in order to put the air back in the tire, you need to use some energy, and you literally need to use a pump. Now, the only pump that we're going to be talking about for a long time will be sodium potassium. We'll introduce it later. We're going to be talking about a lot of ion channels. So I want to give you 4 main categories that you need to know. And the best analogy that you should think of when you think about ion channels is the door. Okay? So ion channels four types that you must know are leaky. So leaky channels, imagine you have a door that opens and closes randomly. When it is open, you can get through. Like today I was trying to use an exit to enter the grocery store. And I should have waited for a person, like it was kind of opening for me, it was random. I couldn't open it myself, so I had to wait, okay? So leaky channels, nothing really controls them, they're just flipping open and closed. Then you have probably the most important, ligand-gated. So ligand-gated channels, they are open when a chemical binds to the channel. Does that make sense? No? Okay. So this door, this one that goes into the prep room, it's closed. Imagine that this is a ligand-gated channel. This dongle is a ligand. So if I go there and I put it next to the lock, it opens. And then, since dongle is not sitting there, when it closes, it closes. Does that make sense? Now, third category is voltage-gated. So in case of voltage-gated channel, check this out, you see? Electricity opens them. Electrical signal opens the channel. We're good? And the last one, which I think we're not going to mention until we start talking about hearing, are mechanically gated channels. So in mechanically gated channels, cell gets deformed, physically deformed, stretched, compressed, twisted. Bent. And it opens the channels. Clear? So you should be able to identify, okay, what kind of channel this stimulus is going to open, what kind of channel this stimulus is going to open. So far, so good. Last little introductory bit before we get to synaptic transmission, which will then be super fast. The concentration of ions. And I often see the signs of terror in students' eyes when I say concentrations. I don't think, maybe I ask you to memorize numbers, maybe 2 times in the course. Know a number, 143. So that's not the case. You don't need to memorize any numbers. This is all you need to know, but this is going to be your, what I call 2 a.m. knowledge. So when you wake up at 2 a.m. and you don't know what your name is, you should know the relative concentrations of ions on 2 sides of the membrane. So if this is the cell, then sodium, calcium, and chloride, their concentration outside are higher than the inside. Am I making sense? And for potassium, it's other way around. Concentration of potassium, in a second, is higher inside the cell than outside. Yes, Katie.
Where is this taking place?
Say it again.
I don't know. Where is this taking place?
This,
Yeah.
every cell in the human body.
During the action potential, or
Say it louder.
the action potential? Or
No. This is a general knowledge, let's call it this way. This is your multiplication table of sorts, So you just always, when we talk about nervous system, literally always, and muscular system, you got to remember, okay, I remember that concentrations of sodium, calcium, and chloride are higher in the extracellular fluid, outside of the cell. Does that make sense? Always, then inside. That's all. And I always remember that concentration of potassium within the cell is much higher than the concentration of potassium outside of the cell. No numbers involved at all. Did I make myself clear? So, we actually have a really good layout. We have all our ion channels. We have our concentrations here. And now we can start talking about what's going on the presynaptic terminal when the action potential arrives. So when action potential arrives, this electrical signal opens calcium channels. How are they gated? Huh? Voltage. Excellent. So action potential opens voltage-gated channels for calcium. Now, knowing that voltage-gated channels for calcium are open, calcium is going to diffuse. Where? Calcium is going to diffuse where? This is diffusion. This is the relative concentrations. Where does calcium diffuse? Into the cell. Does that make sense?
Yeah.
Because the concentration of calcium outside of the cell is higher, and it's going to diffuse towards the lower concentration within the cell. Now, diffusion of calcium, I should have looked up. Oh, I remember, there's an acronym, it's called snare complex, like a drum, snare complex. So calcium activates snare complex. It's not for the exam, trust me. I could barely remember it. The point is, calcium stimulates vesicles within the neuron to start releasing a neurotransmitter. Neurotransmitter. Couple of important things. Well, so far one important thing. The stronger the stimulus, the longer calcium channels are going to stay open. That make sense? The longer they stay open, the more calcium diffuses into the neuron. The more calcium diffuses into the neuron, the more neurotransmitter will be released. So basically, the amount of neurotransmitter that will be released into that synaptic cleft is proportional to the amount of calcium that will diffuse. Does that make sense, that statement? No?
Say it one more time.
Sure. So, the stronger the stimulus, the longer calcium channels are going to stay open. The door opens. It means more calcium will diffuse into the cell. More calcium diffusing into the cell means more neurotransmitter is going to be released into the synaptic cleft. Good? Got it? And then the last step here, really, the last step of synaptic transmission, neurotransmitter binds to the receptors on the postsynaptic neuron. Now, what happens afterwards is going to be a separate story. Like, seriously, separate story. We're going to talk about it separate occasion. So far, this is basically like, that's the transmission is done. When neurotransmitter bound to a postsynaptic neuron, awesome, perfect. Some stuff is going to happen, we'll chat later. Signal is transmitted. We're clear. The more neurotransmitter you have in the synaptic cleft, the stronger the signal. Quite obvious. The longer it stays in the synaptic cleft, the stronger the signal. Does it make sense? I usually compare it to this. So, let's say I'm knocking on the door. That's your neurotransmitter. So neurotransmitter knocking on the door. And like that signal gets really, really annoying after like maybe 3, 4, 5 minutes, right? Stronger signal. Which brings us to the final, really clinically very relevant idea. The action of neurotransmitter has to be terminated. You have to stop the signal. You see what I'm saying? Somebody comes over to a house and pushes the doorbell. Doorbells aren't designed that it's just like, you click on and then it rings and rings and rings and rings and rings. It's going to ring only while person is holding the thumb, right? Right? And if they're not assholes, they just pull it back. So, how can you terminate? One way is the diffusion, and this way sucks. It objectively sucks because diffusion is very slow. It's not a reliable way to get rid of neurotransmitter. That make sense? Another way is to take it back. It's called reuptake. Okay? Also pretty straightforward. And the third way to terminate the actions of neurotransmitter is degradation, enzymatic degradation. Okay? So far we good? Oh, awesome. So before we jump to why it's clinically relevant, I want to say a few words. So first, it kind of reinforces why chemical synapse can transmit signals only one way. Because your dendrites cannot make neurotransmitter and then cannot release it. They just don't have stuff. Okay? And your axonal terminals, they don't have the receptors. They can't receive the signal. That's it. It's one-way street. Make sense? Now, I want to ask you, we're going to talk about some really cool clinical, like real-life stuff. So what if somehow you can inhibit reuptake or degradation? You can't do anything about diffusion, but quite frankly, it's so slow that we can kind of neglect. So if you inhibit reuptake or degradation, what happens to the amount? Listen closely, think. I know the first urge, what is the first urge to say, but if you block reuptake of neurotransmitter or degradation of neurotransmitter, what happens to the amount of neurotransmitter in the cleft? Huh? Absolutely. And if amount of neurotransmitter in the cleft increases, what happens to the signal?
Stronger.
Does that make sense? That's really simple, right? Well, in some folks with anxiety, levels of dopamine and serotonin are decreased. People do not release those happy neurotransmitters in sufficient amounts. Does it make sense to you? So Prozac inhibits the reuptake of serotonin. So amount of serotonin in the synaptic cleft increases, the signal, like the calmness increases. Okay, are you with me? You're familiar with cocaine? I mean, I'm saying, don't get me wrong, but you know what cocaine is.
We're familiar.
Huh? Yeah, like everybody knows. What I'm getting at, you probably know it's kind of a part of popular culture, like memes. People who snort cocaine, how they behave. They're really jittery. Because cocaine is a very strong inhibitor of the reuptake of dopamine and norepinephrine. Norepinephrine that used to be called adrenaline. So what they get is massive amounts of this very excitatory neurotransmitters that make them jittery. That's effect of cocaine. But one of my favorite stories, all of those clinical stories, you can find in the notes, there are plenty of them. But one that really illustrates why we should pay attention to this is the story about inhibitors of the degradation of a neurotransmitter called acetylcholine. There are pesticides called organophosphate pesticides. And in the 1930s, German scientists, Wales, noticed that people, sometimes farmers, get intoxication from those pesticides. And those pesticides lead to a very interesting syndrome that they called sludge syndrome. So pesticides, as we learned later, those pesticides inhibit degradation of acetylcholine. Amount of acetylcholine increases, the signal massively increases. Acetylcholine is really, really important neurotransmitter in regulating your autonomic functions. And sludge stands for salivation, lacrimation, urination, defecation, gastrointestinal cramps, and emesis. Emesis is vomiting. Now, each of these things is not super pleasant. But people had all of them at the same time. Sounds pretty bad, right? So, German scientists ran with it and created sarin, soman, and VX nerve gases. Chemical weapons. Okay? That makes sense? Now, sounds like a funny story. Well, kind of. Why I'm telling you this? I'm going to ask you, what kind of drug? Not the name of the drug, but what drug does. So, there is a drug that soldiers carry in the first aid kit, at least Russian soldiers. I was in ROTC, so I know that. They carry in the first aid kit in case there is a nerve gas attack. So, you have a lot of neurotransmitter that will cause absolutely terrible effects. But, a little hint, to cause these effects, this acetylcholine has to bind to the receptor. So what this drug should do. One step from what I said. You have a person who's poisoned. That person has a lot of acetylcholine here. Okay, degradation doesn't work. Reuptake, acetylcholine doesn't get reuptaken. Okay. Huh?
Bind to the receptor?
It has to bind to the receptor, but what should drug do?
Either get rid of the receptor or bind to it.
Can you say it louder?
Either bind to the receptors for it, or remove the receptors entirely.
Okay, so removing the good, you're practically right. We can't remove the receptor, but we can design a drug that would bind to the receptor itself and block acetylcholine from getting there. Does that make sense? Kind of taking the seat. This drug is called atropine. Okay? It's a real drug, and it was isolated from a plant. I don't know the proper English name. In Russian it's called belladonna, and belladonna in Italian is pretty woman, because women use the juice of that plant that contained atropine this drug also dilates, what's the word? Pupils. And it was considered to be pretty, so they put some of that juice in the eyes. Sometimes they OD'd, which didn't go well. But, you know, that's beauty. So, but yeah, atropine actually is used in optometry now for measurements of certain eye parameters. When they need to completely relax all eye muscles, intrinsic eye muscles, they'll just give you some atropine and your pupils will be dilated and you cannot focus your eye for about a couple of weeks. Okay, that makes sense? So, now, this brings us to two terms that I really want you to know. Atropine was an example of the receptor antagonist. So atropine antagonizes, inhibits acetylcholine receptor. Does that make sense? Just a term. So when you say it inhibits, just antagonist. Now, when people who smoke or who used to smoke, like I used to smoke, smoking is awesome. Like you feel great. It's BS when people say, hey, no. I quit, but it was not easy. So what happens? Nicotine is acetylcholine receptor antagonist. Sorry, agonist, and mixed up the words. Misspoke. Okay, so it's agonist. So nicotine additionally stimulates the receptors in your brain, making, sometimes people say that when after they smoke, they feel smarter, they think more critically, totally possible. So, basically what I want you to understand, what are those terms? So if I ask you in the question, if you have a patient with such and such condition where neurotransmitter, some stuff happens, and ask you, what would you use to treat this condition? Agonist, antagonist, reuptake inhibitor, this, that. Does that make sense? So those questions are kind of hard, but I don't remember if I mentioned this, but I strongly suggest I'm going to bring some scratch paper to the exam, to any exam. I encourage you to do a brain dump. When we start the exam, and all you have on your desks is the pencil, and I bring you Scantron and the actual exam, and you have some scratch paper. At this point, like when I say basically, go, you can put on that scratch paper whatever you want. You can draw the synapse, you can draw the depolarization curve, like classifications. Take your time. Does that make sense? So it's not an open book. But if you retain it here, it's fine with me. And you can use it, that's even better. Are we clear? Now, kind of concluding remarks on neurotransmitters. Yeah, let's do. Very, very few actually. So what do you need to know about neurotransmitters? In the lecture notes, there are, I think, 4 pages with neurotransmitters. There are small molecule. You can write it down. I'm telling you exactly what you need to know. There are small molecule neurotransmitters. There are 2 pages with them. You need to know all the way up until glycine, including glycine. So it will be like acetylcholine, norepinephrine, dopamine, serotonin, histamine, GABA, glutamate, and glycine. I think that's it. Basically, like you go full first page. On the second, I think there are 3 rows. Third row is glycine. You do glycine. That's it. What do you need to know? You need to know that these are small molecule neurotransmitters. Okay? And you need to know whether they are excitatory or inhibitory. Or either. So I can ask you this. Which of the following neurotransmitters is inhibitory? Acetylcholine, norepinephrine, dopamine, GABA. Okay, acetylcholine, norepinephrine, dopamine are all either. They can be either inhibitory or excitatory depending on the context. GABA is exclusively inhibitory neurotransmitter. That's your answer. I can throw in which of the following small molecule neurotransmitters. So you need to figure out which of these is the small molecule neurotransmitter. Clear? Large molecule neurotransmitter. 2 rows. The first one, on the very first slide with large molecule, neuropeptides, is endorphins and enkephalins. They are inhibitory. Second row are tachykinins and substance P. They are excitatory. Period. Nothing else can be on the exam. Am I clear? So I'm not going to ask you about the peptide Y that works in the, or cholecystokinin. Nothing of this is going to be on the exam. Okay? Good? Last bit about neurotransmitters, really straightforward. Which ones are working faster, small or large? Which one will transmit the signal faster, small or large? Small, because they diffuse faster. Okay? That makes sense? I mean, wide receiver runs faster than the lineman. Now, which ones will produce the response that will last longer? Small,
Large.
large. So small, rapid, short response. Large, slow, long-lasting response. We good? Questions? Okay. I'm gonna